this final talk will focus on a group of proteins that you've heard about from Diane Lipscomb and others and Joe Zamponi, high voltage activated calcium channels, which really play a central and indispensable role in the biology of all excitable cells due to the fact that they're bilingual. They speak the language of electrical signals as well as chemical signals. So they open in response to depolarization encoded in action potentials, allows calcium to come inside the cell, and that triggers biological responses. And that paradigm underlies synaptic transmission in the brain, excitation, contraction, coupling in the heart, and release of insulin from the pancreas. And these channels are associated with a number of various diseases. And they're also very important drug targets. Okay, so they come in several different flavors, seven of them. But they all have this common architecture, pore-forming subunit, auxiliary cytosolic beta subunit, and this extracellular alpha-2 delta subunit. They're very important pharmacological targets, but one of the issues with some of the small molecule blockers is that these channels are prevalent everywhere, and so they can have off-target effects due to the expression in different tissues. So one enduring line of research in my lab over the years has been really trying to make genetically encoded calcium channel blockers, which we can direct with tissue specificity. Okay, so at the time I started my lab, around the 2001, a paper came out from Susumu lab where they used yeast-2 hybrid to find binders for calcium channel beta subunits. And they fished out a small G protein called GEM. It turned out to be a member of 4-member families of small G proteins that are of the RAS superfamily. And you've already heard about RAD and its role in adrenergic regulation by Stephen Marks. Well, when they fished these out and co-expressed the channel with the with these RGK proteins, they found that these were actually very robust calcium channel inhibitors. Here's just a reproduction of that work in our own lab here. Here are cells that express a calcium channel. If you co-express REM and come back 24 hours later, the current is totally wiped out. So in a sense, this is a voltage-gated, this is a genetically encoded calcium channel blocker. The problem with these RGKs is that they do multiple things. They regulate the cytoskeleton. They regulate different types of kinases and things. So they would be very, very poor drugs, ultimately. So what we did was to try to figure out how they worked and then try to really mimic their mechanism of action so that we could create our own genetically encoded blockers. And that was work that was undertaken by Ting Yang when she was a graduate student in the lab. And basically what she found, so here's the alpha subunit and here's the beta subunit. And what she found was that the RGK proteins could really inhibit the channel in multiple ways. The first one being removal from the membrane, and the other two really relied on inhibition on the channels on the cell surface itself. Irrespective of those three mechanisms, they all relied on the RGK binding to the beta subunit. So in order to mimic them, the first thing we needed to find were these handles that would bind the beta subunit. And so that's where Travis Morgenstern, when he was in the lab, a graduate student, decided to identify nanobodies to these calcium channel beta subunits. So he purified the beta subunit, sent it to a farm in Massachusetts. They injected a llama and sent him back blood from which he isolated lymphocytes, generated a phage library of the nanobodies, and used phage display together with ELISA to identify nanobody binders to this antigen. Okay. So a nanobody is really a heavy chain only antibody that's produced by things like llamas and sharks. And the nanobody is really the antigen determinant, the antigen binding region that's shown right here. So around 12 to 15 kilodaltons or so. All right, so once we had the nanobody, we had to identify whether it was still binding the beta subunit inside the cytosol, and he used a FRED-based system to do that. There are 4 different types of beta subunits, and what this flow cytometry FRED assay indicated is that all of them, this particular nanobody, which we call F3, is a non-selective beta subunit binder. Okay. So what does it do? So one of the things that we know that the beta subunit does is it binds the calcium channel alpha subunit and it traffics it to the cell surface. So we have a flow cytometry assay that allows us to measure the total expression of the alpha-1C by looking at YFP that's tagged to the channel. And then we look at the surface channels using a surface epitope that we can probe with a fluorescent toxin. And then we subject those cells to, those transfected cells to flow cytometry, either with or without the nanobody expressed. And so what you see here, looking at the red fluorescence indicating the surface, is that in control conditions, you have a lot of YFP positive cells that traffic to the surface. And when you add the nanobody F3, it does absolutely nothing to it. So it doesn't affect the trafficking of the channel, nor the YFP signal. And if you record currents, the nanobody is completely inert. Okay. And so under the best conditions or circumstances, say what we had is a nanobody that binds to this complex but does absolutely nothing. And so now we can address different things to it. And one of the things that we first thought about was to try to make a system that would mimic the ability of RGK proteins to remove the channel from the cell surface. So what we did here was to add the catalytic domain from an E3 ubiquitin lacase, NET42, to the nanobody F3 here. And the idea is that it would bring, the nanobody would bind the beta subunit and bring this HEC domain there and then ubiquitinate the complex and remove it from the cell surface. So does that work? It works really well. So under control conditions, the nanobody itself, you just by itself, you have robust surface expression of the channel. When you have the net4 fused nanobody, that signal is essentially gone. And it's dependent on ubiquitination because if you make a catalytically dead version, you get that surface trafficking back. So if you now look at currents, we're gratified to see that, in fact, the currents are totally wiped out. And so this is a very robust calcium channel inhibitor. There are multiple types of voltage-gated calcium channels that rely on this interaction with the beta subunits. And we found that this molecule was able to block all of them. So we call this cavablator because it inhibits calcium channels based on binding to the beta subunit. And the cartoon here is really what you'd imagine if you cross the Terminator with a camel. Okay. All right. So what is it good for? You have this really robust genetically encoded calcium channel blocker. So one thing that we thought it might be useful is pain, and you've already heard about some wonderful introductions about chronic and neuropathic pain. So I won't go through it, but just the idea that it's a prevalent problem, and there are no real great indications for it, and so that's why there's a lot of efforts to try to develop new blockers or new therapeutics for chronic pain. And so where we thought cavablator might be useful really comes from considering the circuitry that leads to the neuropathic pain. So it starts off from peripheral overactivity of peripheral DRG neurons, nociceptive neurons, and they the signal is conveyed at the dorsal horn of the spinal cord to secondary neurons that projected to the central nervous system to deduce the pain. And so the transfer of signal here is absolutely dependent on these voltage-gated calcium channels. And so the simple idea, is that if we're able to express in these peripheral neurons the cavablator, it would stop that signal from really taking root, and then you would stop the pain in its tracks. Okay. So Chikun Tang, who's here, and Travis set out to test this. So what they did was to made an AAV that encoded cavablator with a TD tomato as a reporter, and they injected that into subcutaneously into the paw of mice, and found that the signal actually showed up in the DRG, the dorsal root ganglia. And these mice also express GCAMP, that's the green signal. So we can then isolate the neurons from the DRG, culture them, and then either patch cells that have cavablator or not. And so if you don't have any cavablator, you have robust currents. With the cavablator, the current is totally wiped out. So it works very well in these DRGs as well. So with that in place, then we turn to a colleague, Guang Yang, and her postdoc, Ling Sun, and they study behavioral models of pain. All right, and so here's the experimental paradigm. We'd inject subcutaneously into mice 28 days before a spared nerve injury model, either a control, AAV9, or one that expresses cavablator. And then do the spared nerve injury and then do behavioral assays after that. So as shown by Murali and others, This SNI model gives you a hyperalgesic phenotype. So the animals experience a long-lasting and deep allodynia, as indicated by this um, decrease in the pore withdrawal threshold in this control condition. And what we found was that in the caviblator mice, they experienced less pain. And so this was due to an injection that was given 30 days. And moving forward, we've seen that the expression lasts for about for months. So the idea here is that you'd probably be able to inject it once and then have pain be long lasting, um, um, decrease for months to years. Okay, and it also happens for thermal as well as cold stimuli. All right, so what was the neural correlates of this? So one of the things that Guang and Linlin did was to put a window near the spinal cord so that we could spy on the activity of the DRG neurons using calcium as a surrogate marker for excitability. So under control conditions, pre-SNI, the neurons are actually quite quiet. 3 days after the SNI, they get more active, and you can see that their activities are actually coordinated. And by 7th days, they're really just going crazy. So that underlies the reason why they have this chronic pain. And what we found was that the in the mice expressing caviblator, those signals were attenuated, showing why the caviblator was effective in these mice. So we were concerned potentially about off-target effects, but we didn't see any with the concentration of drug that we injected subcutaneously, either looking at the open field test or various tests for motor coordination or locomotor functions. Okay. All right. So Gerald Zamponi and his trainee Erica wrote a spotlight on this and came up with a very clever title that I was actually quite jealous of. I wish I'd thought of it myself. The calcium channel terminator, hasta la vista pain. Okay, so that's ultimately what we want. But as you might have noticed, we found that the relief of the pain was partial, right? And so the question that we've been sort of grappling with is how can we maximize this and then really get rid of all the pain, really make it a hasta la vista pain model, as it were. And one of the things that we'd want to do is to increase the amount of caviblator that we express. But caviblator, you can see, if it gets where it doesn't need to get, like in the heart, for example, it'll be very problematic. You do essentially stop the heart. So how do we get hasta la vista pain without hasta la vista patient? That's the model. Okay. So well, one of the things that we could exploit is that there are different beta subunits expressed in different tissues, for example. The heart has beta 2, neurons have others. So could we make a beta isoform selective inhibitor? And that's one of the things that we set out to find. And so in the nanobodies that we got from our injection into the llama, we have dozens of these. And in screening them, we found one, NBE8, that was selective for beta 1, didn't bind any of the other betas. So could we make this into a selective inhibitor for beta 1 bound channels? All right. In doing structure function of this, what we were surprised to find was that, in fact, this nanobody bound the conserved domain of the beta subunits. All these 4 betas have a highly conserved domain. And it was surprising to us that this nanobody actually bound that conserved domain. So we teamed up with Dan Minor and Nihad Nirwan to get a crystal structure of the 2 nanobodies together with the beta subunit. And the beta subunit has this conserved core of the SH3 and the GK that's shown right here. And F3, which bound all the beta subunits, bound this face of the SH3 domain through residues that are conserved amongst all the betas. And NBE8 found a stretch of about 5 residues that were different in beta 1, and that was what it utilized to confer specificity for beta 1. So, unlike NBF3, which was inert when we expressed it with the channel, NBE8 by itself intrinsically inhibits the channel amplitude as well as causes an inactivation of the channel, and it does that only in channels reconstituted with beta 1 and none of the other beta subunits. So this gives a really good proof of concept that you can use these nanobodies to actually make novel channel modulators with unique properties. How does it do this? Well, we found using the flow cytometry assay that it inhibits the trafficking of the channel by about 50%. And in single channel analysis, it also inhibited the open probability of the channel by about 50%. And those two account for the 75% inhibition of the current. We wondered whether we could make it even better by now adding a heck domain of the net4 to it and really make it completely eliminate calcium channels. And we found that, in fact, we did. If you armed it now with a heck domain of net4, now you created this molecule that we call chisel1, calcium channel inhibitor by selectively blocking beta 1. I didn't come up with that. That was Travis. All right, don't blame me. All right, and chisel 1 is a completely, totally eliminates beta 1 currents. And it does it in a way that's highly beta selective. Okay. So far, I've only shown you that it works in hex cells. Does it work in actually native cells? And muscle actually provided us a great context to test efficacy and selectivity. Skeletal muscle expresses alpha-1s together with beta-1. Cardiac muscle expresses alpha-1c with beta-2. And so if chisel-1 is efficacious, it should inhibit the skeletal muscle current. And if it's selective, it shouldn't affect the cardiac current. And that's precisely what we see. It's completely able to eliminate the current in the skeletal muscle, doesn't touch the one in the heart. And so we think that chisels might be a way that we can really amp up the inhibition of pain without killing the animal, because you don't have some of these other betas in the heart, for example. The second way that we could do, how am I doing for time? Oh, okay, that's good, perfect. All right, the other way that we thought about that we could do it is if you had a way to really titrate the amount of inhibition that you had. And if you notice so far, all the inhibitors that I've given you, just like the RGK proteins, are constitutive inhibitors. Is there a way that we could make something that we could turn on and off and then be able to titrate the amount of the inhibition? Okay. So going there, we turn back to how these RGKs were able to inhibit this current. And this second mechanism really depended on two things. The RGK had to bind the beta subunit and also the plasma on the plasma membrane. And we found that out by if you eliminate this C terminus, which targets it to the membrane, you lose the inhibition. But if you're able to put another domain, such as the C1 domain from protein kinase C, which you can dynamically regulate the association with the membrane with a small molecule, then you can make an inducible inhibitor. Okay. And so that led us to think, why do you need those two things to be true? Beta binding and membrane targeting. So this is the hypothesis that we had, that when the RGK binds the beta subunit, which is bound to the 1,2 loop of the channel, and in this configuration, the RGK is not bound to the membrane, and the channel opens, and you can get current influx. Once you add a phorbol ester, it hooks up the C1 domain to the membrane, and it just pulls on the 1,2 in a way that tickles the channel and sort of closes it. Okay, if that was the case, then you didn't really need the RGKs to do that. We should be able to really put the C1 domain directly on the beta subunit and use that as a way to close the channel. And so that's something that we could test, and we did. We put a C1 domain on the C terminus of one of these cytosolic beta 3, so that you can get a PDB-induced translocation to the membrane. And so in channels that are reconstituted with this molecule, which has a C1 domain attached to it, if you add phorbol ester, you get a very rapid and deep inhibitor of the current. So you basically converted the beta subunit from an activator to one that you can inducibly use to close the channel. And we found that, in fact, this was actually very general. You could find other proteins that bound cytosolic parts of the calcium channel. And if you hook them up to the membrane, you could tickle the channel to create inhibitors. So we refer to this as a channel inactivation induced by membrane tethering and associated protein, right? So the CHIMP right here. So as you hook up to the membrane, you close the channel. So the final thing that I want to ask is that could we then use this approach with our nanobodies to now create an inducible inhibitor of the channel? And yes, you can. So there's a nanobody, fuse it to the C1, now you have a way that you can titrate the amount and the kinetics of inhibition. All right. So let me just summarize. I've spoken to you about three things here. Cavablator is a genetically encoded calcium channel inhibitor that we've shown that it can be utilized in vivo to provide long-lasting relief of pain. Spoken about chisels, which give you isoform selectivity. And also this approach CHIMP that allows small molecule control of calf channel inhibition. So we think that the combination of these three will allow us to be able to really get traction into pain. And the other thing I just want to mention is that all of these approaches are not unique to the calcium channel. It can be applied to your favorite channel as well. So I thank the people who did it. Where's Travis? Oh, here's Travis. Papia is not shown here, but she's up here, contributed to that. Arden wasn't able to show his work, but he's done some beautiful work in the T-type channels that will be ready for prime time soon. And Chikung, who partnered with Travis to do this work. Thank you.
And actually, I keep wondering, I keep noticing, I don't work on pain, so I've never seen any of this stuff before, but I see that everybody's getting only partial inhibition by hitting whatever channel they're hitting. And I'm wondering, has anybody tried combining them to see it gives you more, or is the rest of it non-excitability dependent?
Yeah. I mean...
it's a general question. You may not have the
general
answer.
Question, okay. Oh, is it for me, though? Yeah. Okay, all right. All right, I mean, in the case of what we have, the way that we did it was to inject a little bit of cavablator subcutaneously, and then a bit of that is taken up into the DRGs. So if you look at the actual amount of DRGs that are infected by that, it's about 10-15%. So there's still a lot of way to go. But, we are afraid that the more you add, the more of targets that you have. So that's why it's important for us to be able to develop these ways that we can now titrate them in a way that be effective.
Super cool story, super cool acronyms. Chisel, ablator, awesome, amazing. The question I have for you is, why go with beta? Pain.
Thank you. Yeah, it's just and we now have a Chisel 3. Just that beta 1 came along first, and so that work's been done, and that work's just been accepted. So,
okay. Thank you. I think we better stop. We made up 2 minutes in this session. Thank you to all the speakers. That was an excellent session.